My presentation is about photosynthetic supercomplexes and their structure. I will start from thylakoid membrane and photosynthetic supercomplexes. In plants, chloroplast contains thylakoid membrane, which are the site for light-dependent reactions. In thylakoid membrane, there are stacks of discs called grana, which are connected by stroma, thylakoids. As you can see in the picture, these are stacks of discs called grana, and they are connected by the stroma. This is the EM picture of thylakoid membrane and chloroplast, and you can see the stacks of discs again here in high resolution here, and they are connected by stroma. Photosynthetic pigments, for example, photosystem 1 and photosystem 2, are embedded in these membranes. In addition to this, thylakoid membrane encloses a continuous phase called lumen, which is the site for photophosphorylation, which generates energy for the plants. Now about the organization of photosystem 1 in thylakoid membrane. In this picture, you can see the organization of photosystem 2, photosystem 1, cytochrome B6F, and ATPase in thylakoid membrane. And you can see that the photosystem 2 is mainly arranged in the folded part of thylakoid membrane, whereas photosystem 1 is mainly in stromal part of thylakoid membrane, in addition to ATPase and cytochrome B6F, which is randomly distributed all around in thylakoid membrane. One more thing is that this folding is unique for different organisms. This makes it distinguishable from different species. For example, in plants and in algae, the folding is really different, which make them indistinguishable. This is the electron transport chain and generation of energy. Photosystem 2 generates free electrons by splitting of water. These electrons are transferred to photosystem 1 through cytochrome B6F complex, as you can see in this picture. And these electrons are transferred from photosystem 1 to ferredoxin, which are used for reduction of NADP. The energy gradient created by proton gradient is used for generation of ATP, which are further used for dark reactions in photosynthesis. In this slide, I will talk about structure and function of photosystems in thylakoid membrane. This is again the thylakoid membrane, where you can see the photosystems, photosystem 2 here and photosystem 1 arranged, and you can see the structure, how they are arranged in the membrane part. Photosystem 2 is basically a dimer which contains more than 20 subunits and light harvesting complexes. It contains two types of trimers. One is S trimer and other one is M trimer. S trimer is strongly bound to photosystem 2 core, whereas M trimer is readily bound to photosystem 2 core, whereas there are CP26, CP29 and CP26, which are monomeric antenna proteins. All of these proteins are involved in the capturing and transferring of light energy to photosystem 2. This trimer is basically dynamic. It can move from photosystem 2 to photosystem 1 depending upon the light condition. I will talk about it later in my slides. So now let's talk about photosystem 1. Photosystem 1 is also a multimer. It contains more than 15 subunits and light harvesting complex 1. Here you can see the core of photosystem 1, and there's an additional belt of antenna protein just below the core. It contains 4 LHCA proteins. In the core, there are PSIA and PSIB proteins, which form the reaction center for photosystem 1. Now I will talk about straight transition. Plants can receive different light intensities depending upon their habitat and weather conditions. In low light situation, photosystem 2 can form megacomplexes, just something like this. And these megacomplexes can be dissociated when the light intensity is higher. If the light intensity is even more, then these trimers, light harvesting trimers, need to be disattached from photosystem 2. So what happens is that these trimers are phosphorylated and they are aggregated together. These aggregated trimers can be attached with photosystem 1. So this attachment is basically called a state transition. As a result of state transition, the excitation energy is balanced between two photosystems. Okay, now I will talk about the evolution and comparison of photosystem 1. Basically, photosynthesis evolved in cyanobacteria. In cyanobacteria, photosystem 1 is a trimer. It has 3 monomers which are arranged in such a way that light harvesting is maximum. In diatoms, there is no structure for photosystem 1 known yet, but it is a monomer in diatoms, and it contains 3 different types of antenna proteins. Whereas in green algae, photosystem 1 is again a monomer, and it contains an additional belt of antenna protein just below the 4 antenna proteins photosystem 1. In higher plants, photosystem 1 is more specialized. As I discussed before, it contains 4 LHCA proteins just below the core of photosystem 1. Now I will talk about the evolution of photosystem 1. In bacteria, photosystem 1 is basically a trimer. During the evolution, photosystem 1 lost PSA-M and PSA-X subunit to convert it into monomer, and it additionally generated a protein which is PSA-H, which is the site for attachment of LHCA trimer. As you can see here, LHCA trimer is attached here with photosystem 1, which happens during the state transition. And additionally, there was a subunit called PSA-G that was developed. This is the site for attachment of antenna proteins. As you can see here, 4 antenna proteins in photosystem 1 of higher plants. So this is the evolution of photosystem 2 and comparison in different organisms. First of all, again, I will talk with cyanobacteria. Cyanobacteria have additional antenna protein just outside the membrane. They are soluble proteins, and they have special absorption of light intensities which are different from chlorophyll, so they can harvest more energy for the core of photosystem 2. In diatoms, again, the structure of photosystem 2 is unknown, but it is known that photosystem 2 is again a core, is again a dimer, but the arrangement of LHCA protein is not known yet. In green algae, photosystem 2 contains 3 different trimers. One is S trimer, the other one is M trimer, and the next one is N trimer, and it contains 2 monomeric proteins which are LHCb5 and LHCb4. And as you know that in higher plants, the photosystem 2 contains 2 LHC2 trimers and 3 monomeric proteins. Whereas in spruce, there are only 2 light harvesting trimers and 2 monomeric proteins, which gives it just different shape and structure. Thank you.